Good morning, everybody. It's my pleasure to welcome you here today. My name is Sharon Rounds. I'm the Program Director for Advance Rhode Island CTR, and which has been working hard to put on this, I believe, 18th All-State IdeA Symposium, which is pretty impressive. This symposium was planned and by the PIs of all of the Rhode Island IdeA programs, and we're very grateful to them not only for their terrific advice but also for their financial support of this terrific day. This year's theme is chronic diseases, from cells to community. And during this day, we hope to reflect the full spectrum of translational and clinical research in our state. And I also would like to say that we're pleased to have faculty from the Northern New England CTR who are also joining us today and will be giving presentations later this morning. So this is in the hope of eventually developing a more regional event. Our keynotes and lightning talks today highlight the extraordinary breadth of translational science, and this includes emerging insights into cell and molecular pathways, innovative approaches to prevention and care delivery, and research that's grounded in the real lives of people and communities. We're honored to have nationally recognized experts with us today, not only our keynote speakers, but also members of our external advisory committee. We're very pleased to have joining us today. And now it's my honor to introduce a guy who doesn't need much of an introduction. Mukesh Jain, the Dean of the Warren Alpert Medical School, Frank L. Day, Professor of Biology, and the guy who allows us to do this here today. Thank you.
All right, thanks Sharon for that very kind introduction, and good morning everyone. Great to see you and thank you for being here. Really a pleasure to welcome you to the 2025 Emerging Areas of Science Rhode Island IdeA Symposium. So as Sharon said, for the past 18 years, the Rhode Island NIGMS funded Institutional Development Award IdeA program has come together for this annual statewide symposium, a unique tradition that really celebrates research excellence, cultivates a collaborative spirit that really defines our regional scientific community. Each year, this gathering serves also as a powerful reminder of how much we can accomplish together when we have a shared purpose and vision. And we're really pleased to be able to host it at the Warren Alpert Medical School again this year. As Sharon said, our theme today, chronic diseases from cells to community, will speak to the breadth and depth of translational power of our community, from molecular underpinnings of disease to real-world strengths that improve health outcomes across populations. So the IdeA programs have been absolutely critical in building research infrastructure and supporting investigators throughout Rhode Island. Advance-CTR has served as a statewide hub for clinical and translational science, delivering core resources, training, and services that have enabled researchers to elevate their work. And in parallel, COBRA awards have helped to establish and sustain interdisciplinary centers of excellence across the state, including at our affiliated hospital partners. And we're also very proud of our collaboration in Rhode Island's IdeA Network for Biomedical Research Excellence, or INBRE, led by Dr. Bong-Suk Choi, who's in the audience. Really an outstanding example of cross-institutional collaboration that is helping us build this critical scientific mass necessary for sustained impact from a small but really mighty state. So all of this work, really your work, is particularly vital in today's uncertain funding climate. Federal support for research is under a strain, making the role of the IDEA program more essential than ever, because the IDEA program ensures that talented investigators in historically underfunded state like ours have access to critical infrastructure, mentorship resources, so that we can contribute fully to the national biomedical research enterprise. It's an incredibly important program. It's a program worth protecting and amplifying. I'd also like to take a minute to recognize and thank today's distinguished keynote speakers. The first who has landed and is en route to be here, so she'll be here very shortly, is Dr. Vivian Chung, who will begin our day by introducing the Human RNome Project, sequel to the Human Genome Project that has its foundings at Brown University. And she'll highlight how RNA modifications influence gene expression and contribute to human disease. And then later we'll hear from Dr. Donald Neese, University of Colorado, who'll speak on the vital role of engaging patients and primary care networks in the prevention and management of chronic illness. We're grateful for their presence, grateful for their insights and inspiration that they will undoubtedly bring to today's program. We're also deeply grateful to Senator Jack Reed for his tireless advocacy on behalf of the IDEA program and Rhode Island's broader research community. His leadership has helped ensure that innovation continues to flourish here. I want to conclude by also thanking our planning team, especially our Advance-CTR team, for all others, as Sharon said, our partners who have contributed, and to our leaders from URI. I see Dr. Carrie LaPlante in the audience, as well as from our hospital systems, Dr. Michael Wagner, present as well in the back. Thank you both for being here. And finally, thanks to all our presenters, lightning talk speakers, that's a cool term, poster participants, and visiting colleagues from Maine Health and the University of Vermont who joined us for the first time. I have no doubt that today's symposium will bring new ideas, strengthen existing connections, and spark new ones. And with that, I am going to turn it over to Dr. Ed Horrid to introduce our congressional leaders.
Good morning, everyone. My name is Ed Hallrut, and I'm honored to serve as the Alva Olway Professor of Molecular Biology, Cell Biology, and Biochemistry in the Division of Biology and Medicine here at Brown University. I also have the privilege of serving as the program coordinator for Rhode Island Advance-CTR, and we're in year 9 right now and getting ready for our renewal application. It's my pleasure to introduce some pre-recorded remarks that we have from the senior senator from Rhode Island, Jack Reed. Senator Reed has long recognized that investing in scientific research is a critically important investment for our community here in Rhode Island. His strong support for the IDEA program has empowered Rhode Island's researchers and institutions to tackle complex biomedical challenges while building a resilient, innovation-driven economy. As you've just heard from Dean Jane, the IDEA Institutional Development Award Program is particularly significant to Rhode Island. Through it, the National Institute of General Medical Science supports biomedical research across historically underfunded regions, enabling institutions such as Brown University, the University of Rhode Island, and the state's health systems to more fully engage in cutting-edge research and development to facilitate training for the next generation of scientists and to stimulate, not least of which, the local economy. Senator Reed has been a key partner as we move to transform our state into a burgeoning life science hub. Because the U.S. Senate is in session today, Senator cannot be with us in person, but we are pleased to provide these video remarks from Senator Jack Reed.
Hello, everyone, and thank you for inviting me to participate in today's program. Let me thank the students and faculty at the Warren Alpert Medical School who are working to advance biomedical research in Rhode Island. You all know that research funded through the National Institutes of Health is a key driver in helping us to better understand, diagnose, and treat life-threatening diseases. But as we seek to advance medical breakthroughs, we need to recognize that good ideas don't come from a handful of institutions. We need to foster innovation in every corner of the country. That's why we created the Institutional Development Award, better known as IDEA, under the NIH. IDEA increases access to NIH funding in states that have historically received low levels of NIH investment. In Rhode Island, IDEA has really paid off, helping our state to become a biomedical research hub. This kind of success is why I work in my role as a member of the Appropriations Committee to push for greater investment in NIH and IDEA. And it's why I am fighting against sharp spending cuts that will impact these programs. In fact, this year, President Trump has launched an all-out attack on biomedical research and academia. I am fighting back against these attacks. We have to protect the United States' role as a global leader in medical research and innovation, and the administration's policies would undermine years of work towards that great effort. Thankfully, all of you in this room have been instrumental to the research and data that helps us make the case for investing in these programs. Your work shows that investment in biomedical research pays off in cures and new treatments for diseases, in a new generation of researchers, and in economic growth and jobs at all levels. Again, thank you to the Warren Alpert Medical School for your important work, and I look forward to seeing the great advances and discoveries you make. Thank you.
Next, we're fortunate today to have a special message from Congressman Gabe Amo. As many of you know, Congressman Amo represents Rhode Island's 1st District and brings a unique background in public service, community engagement, and policy leadership at the highest levels of government. A proud Rhode Islander, he serves on the House Committee on Science, Space, and Technology, where he has been a tireless champion for biomedical research. We are grateful for his support and for taking the time today to share these thoughts with us.
Hi everyone, it's Congressman Gabe Amo here. I'd like to welcome you all to the 2025 Emergent Areas of Science Ideas Symposium at Brown University's Warren Alpert Medical School. I truly wish I could be there with you in person, so I wanted to pass along my best wishes as you come together today to celebrate biomedical research in our great state of Rhode Island. And in a state like ours, there is so very much to celebrate. The RI IdeA network of biomedical research excellence has helped propel research capacity at Rhode Island institutions, leading to major developments in so many key areas like cancer treatment, environmental health sciences, and neuroscience research. Biomedical research not only drives improvements in health outcomes, but it also provides substantial, very meaningful downstream economic benefits. These innovations do major things like lower healthcare costs, increase workforce productivity, and extend life expectancy. But unfortunately, this incredible work is under fierce attack. The Trump administration has politicized the work of the National Institutes of Health, threatening funding and even terminating 2,100 research grants valued at more than $12 billion. And just last week, we saw Health and Human Services Secretary Robert F. Kennedy Jr. fire the entire vaccine board of experts. This radical decision leaves Americans vulnerable to COVID, the flu, or many other future health outbreaks. But Democrats are firing on all cylinders to fight back. We're supporting litigation, we're introducing legislation, we are demanding answers from Trump, and we're rallying support from people just like you. I led over 150 of my Democratic colleagues in opposing the NIH's illegal decision to reduce the reimbursement rate for indirect research costs. And thankfully, a federal judge has stepped in to extend the temporary restraining order, stopping the Trump administration's NIH leadership from slashing critical research funding. I'm proud of Rhode Island's own, the Attorney General Peter Nerone and for leading a coalition of 23 attorneys general in preventing HHS from unlawfully cutting nearly $11 billion in essential public health grants nationwide, including $31 million for Rhode Island. Thanks to the actions of attorneys general, a judge issued a temporary restraining order to prevent the termination of this funding for the time being. But I'm not going to stand idly by while the Trump administration slashes public health funding and congressional Republicans ram through a dangerous, ugly bill that would rip away health insurance from 16 million Americans. I really just want to say thank you all for being here today to support Rhode Island's biomedical research. And thank you to the Warren Alpert Medical School for organizing this symposium. It's important that you are gathered. I welcome your thoughts, your ideas, your passion, and your energy. And I look forward to all that we will do together. Have a great symposium.
It's great to have that support from our congressional delegation. And now I turn the podium back over to Dean Jane.
All right, I know the crowd's on the edge of their seats. Dr. Chung is in the house. So real pleasure to introduce our first keynote speaker, Vivian Chung. So Dr. Chung earned her bachelor's from UCLA, MD from Tufts University, pediatrics training and postdoctoral fellowship at UCLA, and neurology training at the Children's Hospital of Philadelphia at the University of Pennsylvania. She has held endowed professorships at the University of Pennsylvania and the University of Michigan, and now joined Brown in November of 2024. Dr. Chung's research focuses on genetic mechanisms of disease, particularly those driven by alterations in RNA processing and gene expression. Her group made the unexpected discovery that RNA sequences can differ from their corresponding DNA templates. That these differences are further explained by RNA modifications or decorations on RNA that you'll hear a bit about shortly. As a physician scientist, she applies insights from this fundamental research to advance understanding of neurological disorders ranging from juvenile onset ALS to Alzheimer's. I would just add, having followed her work for well over a decade now, that the creativity, the thematic consistency, and the investigative breadth and depth with far-reaching theoretical and practical implications of Vivian's scientific contributions are truly exceptional. Beyond research, she has provided national leadership in science, having served as president of the oldest physician scientist organization, the American Society for Clinical Investigation, and she was the inspiration and driving force for the 2024 National Academies of Science, Engineering, and Medicine report entitled Charting a Future for Sequencing RNA and Its Modifications. A dedicated advocate for diversity in the biomedical workforce, she has published op-eds in the Washington Post, New England Journal of Medicine, and JAMA, to name a few. Her mentorship has shaped the next generation of scientists, with her trainees earning prestigious honors, including Rhodes Scholarships and the Harold Amos Medical Faculty Development Award. And for her numerous contributions to science, medicine, and clinical care, it will not surprise the audience that she is a recipient of numerous honors and awards. I will not enumerate all of them. She gets very upset if I do that. But I'll just note the Kurt Stern Award from the American Society of Human Genetics, and she's an elected member to the National Academy of Medicine. With that, Vivian.
All right, thank you. I'm actually just a dog lover and a pediatrician. So I'm really glad to be here today. It's an honor to be with all of you this morning, and not just I want to thank all of you, not just for inviting me here today, but for your unwavering dedication to advancing biomedical research across the country. The IdeA program is bold and necessary. It recognizes that talent is everywhere, but opportunity is not. by investing for research excellence in states that are historically underrepresented by the NIH funding, IDEA is helping to ensure that discovery, innovation, and the benefits of science reach all corners of America. And as someone who spent most of my career at the interface between science and medicine, I know how important this is. So today we stand at a turning point in biology, one that is shaping how we diagnose, treat, and even think about diseases. At the center of this transformation is RNA. We are beginning to understand that not only the genetic code, but the language of RNA, the dynamic, chemically modified molecules that carry out and regulate what our genes instruct. So just as the Human Genome Project transformed our understanding of DNA, we now need a Human RNome Project to help to map, define, and harness the power of RNA. This transformation must happen in places like Rhode Island and the IdeA network. The next generation of breakthroughs, whether it's in Alzheimer's disease or in cancer or in genetic diseases, will come from better technology and broader participation, new voices, regions, and ideas. So thank you for everything you do to bring these voices into the conversations and into the laboratories. So what I'd like to do today is to share with you some of the work that my team and I are doing to turn RNA discoveries into treatments and explore how we can work together. In 1993, scientists discovered a gene variant called ApoE4 that increases one's risk of developing Alzheimer's disease. Today, we know that more than 2/3 of Alzheimer's patients carry this genetic risk factor. Yet after 30 years of research, we still do not have a treatment that can prevent people with this risk factor from developing Alzheimer's disease. But I'm hopeful that it's about to change. In the last decade, we have seen remarkable advances, new medicine for cancer, new treatments for genetic diseases, computing power have exploded, and these technological leaps are now accelerating progress for diseases just like Alzheimer's. To appreciate the potential of RNA in reshaping how we treat and talk about diseases like Alzheimer's disease, I'd like to begin with two stories. These are real stories. I have just changed some of the details and names, so it's not so clear who these people are. Yeah. So Annie, thank you so much. So Annie was born to Terry and Tom, who were really proud to bring a daughter into their family. And her 10-year-old brother was very excited to have a little sister. But within a few months, Terry noticed something was not quite right. Annie seemed unusually floppy. She struggled to lift up her head and was not meeting her developmental milestones. After many visits to doctors and specialists, Annie was diagnosed with spinal muscular atrophy, a devastating genetic disease that just 10 years ago, most of the patients died before the age of 3. But today, thanks to an RNA-based medicine called nusinersen, Annie is thriving. In kindergarten, she ran and played with other kids. Now in first grade, she loves reading. She builds Lego castle with her brother and aspires to become a tornado chaser. True. Nusinersen works by modifying RNA to compensate for the genetic defect in DNA that causes SMA. It allows patients like Annie to have the SMN protein for the proper function of her nerves and muscles. I have one more story to share. So John is a farmer from rural Tennessee. Heart disease runs deep in his family. His father, grandfather, aunts, and uncles all died young from heart diseases and strokes. But despite his best effort with diet, exercises, and medications, John's cholesterol level remained dangerously high. But living in a remote area, it was hard for him to go to see doctors regularly or keep up with daily prescriptions. But that changed with inclisiran, an RNA medicines that lowers cholesterol by silencing the gene PCSK9. With just 2 injections a year, John's LDL cholesterol is now well controlled. He takes the 1.5-hour drive every 6 months to see his cardiologist for his injection and checkups. The rest of the time, he focuses on growing his soybean farm. These stories are not just about rare diseases or heart conditions. They are proof that RNA medicine can precisely target genes to treat the root causes of diseases safely, effectively, and sustainably. But they also raise a larger question. What else could we do if we understood RNA more completely? But before I go on, I'm going to give all of you a quiz. Can anyone tell me what this is? Speak up. I actually have an award for this, so anyone. Yeah, please.
National anthem?
Yes, excellent. So these are indeed the opening lines of the Star Spangled Banner. And all I did was to remove some letters. Suddenly it becomes hard to recognize, even though it's a song that most of us know by heart. And this is how we are currently reading RNA sequences. For decades, we have worked under the assumptions that RNAs are just like DNA and have 4 bases. While that is true when it is being synthesized, within 1 second of synthesis, the RNA is extensively chemically modified to and now we know there are over 180 modifications and probably many more yet to be defined. These modifications are not random. They are instructions. They tell the RNA how to fold, when to be translated, when to be degraded. If we try to read RNA without accounting for these modifications, and we are just like trying to sing a song with words missing. And this is why we need a human RNA project. Just as the Human Genome Project gives us the blueprint of life, the Human RNA Project will decode the active language of RNA. In RNA, with more than these 180 modifications that are part of the RNA sequence, they tell the cells when to be translated, where to go, how to fold, what to do, when to be erased. RNA is not just a messenger, it is a regulator, a sensor, and a switch. It really is biology's multi-tool. Yet we still lack the standards and the tools to read RNA in its full complexity. Today, we lack the ability to reliably sequence RNA and identify this full set of modifications, let alone to understand its function. And this is why we launched the Human RNA Project to sequence RNA with its modifications, to develop technologies that can detect, interpret them, and to create a dictionary that links RNA chemistry to its biological functions. Just like the Human Genome Project, this work will have ripple effect through society and science. In medicine, the RNA will uncover drug targets and therapeutic strategies, especially for diseases like ALS, Alzheimer's, and cancer, where gene regulation and cellular stress responses are disrupted. In agriculture, RNA modification will help us to engineer better crops and safer pesticides. In data storage, RNA molecules, programmable and information-rich, could serve as a medium for archiving digital information at massive scale. So just as the Human Genome Project helped train the whole generation of genomic scientists, the Human RNome Project will develop the next generation of RNA biologists, chemists, computational scientists. This is the foundation of what we call the biofuture. The new era of science that is information-rich and RNA-centered and grounded in US innovation. But it is also an opportunity for regions like Rhode Island and every IDEA state. Because building the RNome requires more than just sequencing machines, it needs chemists, biologists, engineers, and entrepreneurs. It needs a workforce trained in the next generation of science and technology and the infrastructure for biomanufacturing and discovery. I believe the IDEA state can lead in this new era. You have proven that when opportunity meets talent, great science follows. So the RNome Project is our next frontier. I hope all of us can work together to make a bright future for that project. So the potential of RNA is vast, but to realize it, we also need to demonstrate that new understanding can translate into medicines. That's exactly what my team and I are doing in our research laboratory on Alzheimer's disease. Alzheimer's disease affects over 6 million Americans. Behind each number is a family with memories fading, loved ones struggling to hold on. It's a disease that strikes at the core of who we are. And yet for too long, we have had too few tools to fight back. But today, we finally may have a way forward. We have one that targets the ApoE4 gene to be sure that the disease-causing ApoE4 protein is not made. For decades, Alzheimer's research have focused on amyloid plaques, these sticky protein clumps that build up in the brain. Many treatments have tried to remove those plaques, but even when successful, they have not done much to slow or stop the disease. Why? Because by the time those plaques accumulate, a lot of the damages has already been done. It is like trying to clean up smoke without putting out the fire. So we begin by asking, what is fueling the fire? What causes the brain to start the spiral to begin with? That question led us to the ApoE gene. We all carry some form of the ApoE gene, which makes the ApoE protein that transport cholesterol and fat throughout the body, including our brain cells, to keep our brain healthy and to repair damages. But one form of the ApoE gene is ApoE4. We inherit one copy of the ApoE gene from our moms, another from our dads. And there are three common types of ApoE, E2, E3, and E4. The ApoE4 is the one that is linked to increased risk of Alzheimer's disease. For about 20% of people in this room have one copy of ApoE4, and 1 in 50 have 2 copies of ApoE4. So if you have one copy of ApoE4, your risk of developing Alzheimer's is increased by about 3-fold. And if you have 2 copies, your risk of developing Alzheimer's is increased by over 10-fold. And strikingly, up to 70% of Alzheimer's patients carry at least one copy of the E4 protein gene. Research have shown that the ApoE protein takes us on a slightly different shape than the E2, E3 protein, subtle but important differences. It is not as effective in transporting cholesterol, and sometimes it is toxic to the neurons, including by causing inflammation in the brain. So here we have a major risk factor identified 30 years ago, and yet we still do not have treatments that targets it. Mukesh know that I never have a good sense of direction. I still get lost all the time, but I never have a copy of a paper map with me when I needed one. But then smartphone came along, and all I need to do is to follow that little blue dot on the map, and now everything changed. The roads did not change, but now I finally know where I am and where I needed to go, or how I needed to get there. That's how the Human Arnome Project works. It offers biology not a new destination. It gave us a clearer path to reach it. We have known that ApoE4 protein, the ApoE4 gene, is a major risk factor for Alzheimer's. But how to modulate its expression remained unknown until we began to look at the RNA landscape around that gene. So a few years ago, my lab and collaborator discovered something that changed how we think about ApoE. We found a special RNA, a non-coding RNA, that sits right next to the ApoE gene. This RNA does not make a protein, yet it plays a critical role in controlling how much ApoE is produced. We called it ANCR, spelled AANCR, and short for ApoE activating non-coding RNA. Think of ANCR as a dimmer switch. When it is on, it tells the cells to make ApoE. When it is off, it tells the cells not to make any ApoE, and ApoE level drops. Why is this exciting? Well, we finally have a way to control ApoE productions. Now we can develop ways to ensure that people with 1 or 2 copy of ApoE4 do not make any ApoE4 protein from the gene. One can also ask, why was ANCR missing all these years when it is hiding in plain sight? There have been a lot of hints saying that there's something interesting in this region. Genetic signals have shown that there may be something that alters Alzheimer's risk. But people were not looking for an RNA, and it turns out that AANCR or ANCR is a regulatory RNA that does not code for protein, and is a very fancy RNA. So remember the chemical modifications that I mentioned? Well, ANCR is regulated by them. So first, it comes in 2 forms. In some cells, ANCR is folded in what is called an R loop. The stability of this R loop is determined by not 1, but 2 chemical modification steps. In that case, ANCR is folded and is off, so no ApoE is made. In some other cells, such as your liver cells or your astrocytes, ANCR is making, a lot of ANCR is made. It is not folded, it is not modified, it's made into a full-length RNA that goes to tell the cells, hey, make more ApoE, and it does. So given this complexity, perhaps it's not surprising that it has remained unknown for this long. The discovery of what controls ApoEs is indeed important, but we did not stop there. In our recent work published recently, we asked what happened if we shut off ANCR. So using human astrocytes and microglia, the 2 brain cell types that make ApoE, we employed antisense oligonucleotide or ASO. These are custom-made nucleic acid that binds to RNA and tells cells to destroy them. The result, ANCR level drops, ApoE level drops, and more importantly, the inflammatory response of the brain cell also decreased significantly. This is key because inflammation is a major driver of neurodegeneration. So thanks to recent support from the Rotary Club, from the CART Fund, we're now launching the next step of this project. We're taking a 2-step approach. We're designing ASO to shut down the ANCR that corresponds to ApoE4 to make sure no ApoE4 RNA is made. And just in case any of it leak through, we are also targeting ApoE4 by sparing ApoE3 RNA. And so as a result, someone who is heterozygous E3, E4 will continue to make ApoE3 protein, but will not be making any ApoE4. And this is what makes our approach different. We're not editing DNA. We're not eliminating R ApoE. We're using RNA-based precision to dial down the key driver of Alzheimer's disease. To target ANSWER and ApoE4, we will use antisense oligonucleotide, this custom piece of genetic material that finds and binds to specific RNA messages inside the cells. When an ASO binds to a harmful RNA message, it acts like a magnet for the cell cleanup system. It leads to the RNA message being destroyed, essentially erased, so it cannot be used to make proteins. So I hope the next time when I return to this symposium, I will report that we have successfully silenced ApoE4 and found that we have reduced inflammation and returned the neurons to a healthier state. And at that point, we will move into a preclinical trial. All right, I'm going to spend like the next 5 minutes being a pediatrician and tell you about a disease that I have been studying for the last decade. So juvenile ALS, it's a very rare disease due to, in this case, mutation in a gene called synataxin. It's an RNA DNA helicase that resolves those R loops that I mentioned earlier. And the presentation is very different from classic ALS. A typical patient would be a young teenage boy who had been playing baseball all his life and suddenly realized he's running more slowly. Or a few weeks ago, I saw a girl who had been playing violin since she was 5 and suddenly realized that her hands are getting weaker. And it's a very slowly progressive disease. Usually patients become, say, a walker dependent or wheelchair dependent in their 40s and 50s. Shown here is a very large family in Maryland with this disease, but there are also a lot of sporadic cases. When I met this family now almost 10 years ago, they told me that since they were in Maryland near NIH, they were very aware of all the genetic discoveries and all the excitement around genetics in the 80s. So they started contacting doctors to get their genes mapped. And so when I met them, they said to me, well, my gene have been mapped for 15 years, but we know nothing about the gene, and we certainly don't have a treatment. So the first thing we did was to ask, so what is the molecular effect of this mutation, and why does it cause a motor neuron disease? And luckily, also at the NIH, at NIAID, and with Susan Gottesman, they have developed an antibody that can recognize R loops. So we used this S9.6 antibody and tested it again for R loop abundance in the different cells from the patients and their unaffected family members. What we found uniformly is that the patients have fewer R loops than their controls. And so we know this is consistent with the autosomal dominant inheritance of the disease, that there is a gain of helicase function leading to the molecular defects. But how does it cause a motor neuron disease, right? I'm going to introduce you to the gene BAMBI. So as I said, the patients have fewer R loops, and R loops form at the promoter of the gene BAMBI. So in the patients, because their synataxin is working too well, it's continuously removing those R loops. So the promoter is in double-stranded DNA, while the unaffected control has an R loop there. And proteins such as DNMT1 that binds to double-stranded DNA will bind to the patient Bambi promoter, but not to the unaffected individual who has an R loop. And as a result, the patient's Bambi promoter is methylated and the gene is silenced. And shown here are sequencing as well as PCR results showing that in the patients, the Bambi gene expression is indeed significantly lower. So who is Bambi? Bambi is a TGF-beta pseudoreceptor. So in most of us, the Bambi promoter is binding to the ligand to modulate the amount of TGF-beta signaling. But in the patients, since they don't have Bambi, everything goes through the TGF-beta pathway, leading to an activation of the TGF-beta pathway. And when we sequence the motor neuron, the iPSC-derived motor neuron from the patients, we indeed see by pathway analysis a very significant activation of the TGF-beta. And to ask what is the effect on motor neuron, we added TGF-beta back to the iPSC-derived motor neurons. And what we found was that indeed there are fewer branches and the neurons are shorter. So also consistent with the clinical findings that we have, that the patient usually show up with distal weakness first because their neurons just cannot go from their spine all the way to their big toe. As a promise to the patient, we have been working really hard to find treatments. So as I mentioned, the patient have one copy of normal centotaxin and one mutated copy. We know that we cannot silence the gene because patients with 2 copies of mutation have another neurological disease called ataxia ocular apraxia. So our goal in this case, so the patient have a C mutation. Our job is to silence the C copy, but leave the T copy intact. So we started developing all sort of siRNA to walk along the gene to find one that will silence the C copy, but leaving the T copy alone. That took us almost 8 years of screening to find the right one. We have to add other mismatches and so on. But now we do have such siRNA in hand that will knock down the C copy. And by sequencing, we can show that the T copy is intact. So patients have actually 50% shown here of kind of the centotaxin protein. And most importantly, we were able to rescue the R loop phenotypes. Instead of having too few R loops, we are now able to increase the R loop abundance at Bambi and genome-wide. So now we're trying to take this up into a clinical grade, RNA therapeutics. At the same time, we also need biomarkers to kind of track the progression and hopefully improvement of that progression. So one of the things that we use is by imaging to measure muscle loss. So when one loses muscle, you get a fat replacement. So by MRI and DEXA scan, we can quantify the amount of muscle loss. And so we're hoping that we can decrease the amount of muscle loss with an RNA therapy. All right, so let me just end by saying that science takes time and progress is almost never linear. But with programs like IdeA, which invest in talent and bold ideas across every region, we are not just imagining what is possible, we are building them. So from life-changing RNA therapies to a national initiative to decode the RNome, we are entering a new era of biology, one that is programmable, bold, and full of promise. The future will not be built by a few institutions alone. It will take new partners, new leaders, and new infrastructures rooted in communities across the country. That's the spirit of IdeA, and that's the foundation of a biofuture. So thank you for your commitment, your curiosity, and your collaboration. I'm honored to be here, and I can't wait to see what all of you can build together. Thank you.
Just wow. Another round of applause. I mean, some of your talks, especially the Alzheimer's, just hit home because at some point, right, this has hurt some of us and definitely friends and family. Any questions, please? Of course.
The question about the neurological specificity of mutations in synaptopsin.
Yeah.
Is there anything in particular about the genes that axon that lend itself to this neurological phenotype?
Yeah, so it's happened, I think it's actually in very fast growing cells. So the patient actually have two other symptoms. Many of them have adenocarcinoma of the colon, and they have a very unusual skin disorder where the collagen are pushing up on the skin. So it seems that the TGF-beta activation is particularly bad for cells that are growing very fast.
I have a similar question. Sorry, can I wake up? With the TGF-beta overactivation, as a lung doctor, of course, I'm wondering, is there an increased frequency of pulmonary fibrosis or hepatic fibrosis? You mentioned skin disorders, and might this possibly be playing a role in those idiopathic conditions?
Yes, definitely. We certainly see not just hepatic fibrosis, but we certainly see pulmonary fibrosis in the patients. Since it's just such a slow disease, right? Unfortunately, most of the patients pass from cancer or some pulmonary issues. And even though it's a neurologic disease that completely spare the bulbar muscle, so there's no problem with them swallowing and breathing from the neurologic point of view, but from the fibrosis point of view, it's certainly an issue. Yeah.
Thank you. Stephen Thomas, University of Maryland School of Public Health. I direct the Center for Health Equity there, and I've been on the advisory board for IdeA. So I just want to echo the accolades and observations that you've made. I've seen that over the past 10 years serving on this board. I followed your talk, and there were places where I said, that's another language. And so there was a word you said, I want to make sure that I'm clear.
Please.
I heard you say the word rotary.
Yes.
And because you said the word rotary at a time when our funding in the national infrastructures under threat, we've also been thinking about alternative partners.
Absolutely.
And I've never thought about the Rotary. And every time I come into a town, I see this sign that says Rotary, that says Lions Club. I see all those in every little town, and I have never reached out to them. And so you've inspired me today to think differently. Can you say a little bit more about how you found the Rotary,
I thought no one would
why
ask.
we should talk to the Rotary, and why NIH may not be the only place that can support our work?
Absolutely. So I just came back from receiving this award from the Rotary Club. Many of you have heard and seen those little blue buckets at grocery stores, coins for Alzheimer's disease. It was started by Mr. Roger Ackerman 30 years ago with the idea that people will go to the Rotary Club meetings and they will empty their pockets of coins. And every year they will raise money and spend the entire amount. This year they raised over $2.3 million from not just Rotary Club across the nation. They are just Rotary Club, mostly from the southeast part of the Rotary Clubs. And they just gave our group $500,000 to develop this Alzheimer's disease treatments. And they nothing against the NIH. I love the NIH. I see patients at the NIH. But it was so nice to be at the Rotary Club. They invited us to their annual meeting. They took us out to dinner. One of the family is going to invite me for a weekend to South Carolina to meet their grandma. I think it really does matter, right? Because sometimes we're all human, right? And in order for us to see the patients and understand what is impacting them, hopefully it will drive our science and not just about a paper or some awards, right? But about that grandma. So it was an amazing opportunity. I do believe that we have to diversify our income sources, and we also can experience. So one more thing about this award, it only takes 6 weeks from the application to the notification. And like in week 4, I got an email from this gentleman saying, "Oh, we're so sorry that we're being delayed." I'm like, wait, what? And it's only 6 pages. And then, like, it's really a phenomenal organization. I encourage all of you to look into it.
Thank you. We have another question up here.
Could you talk a little bit about how or whether these RNA therapies can be leveraged for in utero treatment?
For
In utero treatment? Like,
yeah, I think that RNA can be used for many things, and it certainly will be easier than to deliver a protein for in utero, and it certainly is safer than changing the genetic code. So, because if something goes wrong, right, you can stop it. And in utero, and you can dose it differently in utero and as a child grows up. So I think there's a lot of potential there. All right.
Thank you again. Questions?
Yeah.
Not a question, but a comment. Just wanted to mention a Rhode Island connection to your work. Christopher Grunzeig was an undergraduate
Yes,
here
I know.
in biology. So I just wanted to call him out, making a contribution to.
Yes, definitely. And I see him all the time. I think there's one last question over there, right? Yeah. Yeah.
Assistant Team G, Assistant Professor at URI. I was just wondering when your RNA treatment is ready and to go for testing either in mouse or human. Kind of what are your thoughts on the delivery route? Thinking about like local delivery, or because I'm sure you know better than I do that the blood-brain barrier is
Yeah.
very big challenge.
Yes, I think we're a little lucky here. I hope that we're a little lucky here. ApoE happened to be a protein that can cross the blood-brain barrier, and it has a receptor signaling part. So we are hoping that we're going to connect that part with an ASO and take it into the brain. Yeah.
Thank you.
Yeah. It's a major issue since we want to give people a lifetime of this treatment. So, yeah.
Thank you for the great talk, Bong-Suk Cho from URI. You show one of those slides, 180 different modification of RNA, much more complicated than DNA. So what are the challenges for sequencing this? And then where you stand on RNome project at the moment?
Yeah. So it is very hard. Right now there's Oxford Nanopore technology that can read maybe 6 to 8 of the 180 letters. So we have a long way to go. I think we will need a new technology. But despite all the challenges with funding, we've been getting very good support for the Human RNome project, both from the NIH and other agencies. I'm definitely out there pounding the pavement to make sure that we understand the importance, right? Not just for biomedicine, but for agriculture, national security, and for biomanufacturing. A state like Rhode Island should be the one that's making the next generation of RNA medicine and pesticides. So I'm very hopeful that we will have a human RNome project launch next year. We have a consortium that includes over 20 countries, and we already have workshops kind of training scientists on how to prepare the RNA, how to do the sequencing. Yeah, all right. One last.
Okay, all right, sorry. You started clapping, so I was like, oh, I don't have to talk. All right, I'll be quick. Thanks. Rusty McCullough, University of Nebraska. Really great talk, really interesting. One of the things that I do a lot of regulatory at my home institution, and one of the constant challenges is that valley of death as you are moving through an IND into phase one and subsequent trials. What are and that's very different from much of the research that you do. It's an entirely different regulatory landscape and support and team. What do you have? What do you need? What do you wish you had in this regard? And where does something like the CTR fit in that equation?
Yes, absolutely. Having a CTR would be amazing. I think the other thing that we need, right, is an understanding that if all the components of an RNA therapeutics are the same, and we're just changing the spelling, then it should go through a very different approval process. I think as I said, and I purposely said, it took us 8 years to find that siRNA for the kids with ALS. We need a better way that's truly programmable and scalable. And I'm actually moving to later on this week to another meeting where I'm hoping to convince a large organization to start thinking about a programmable, scalable RNA therapeutic platform. Yeah.
I have the mic again.
That's fine.
So, I work in the area of bringing populations into clinical trials that have been historically underrepresented. You mentioned Alzheimer's. If I have the numbers right, African Americans have one of the highest Alzheimer's rates in the country.
Yep.
Let's talk about the country. So here's my question. Does it matter where the DNA comes from? When we see these variations in patterns in disease that flow along racial lines, even though race is not a biologic, all the deals, and yet we see this pattern.
Yep.
So is it important to have African Americans in Alzheimer's research studies, or does it matter where the data comes from to solve this problem?
It's critical because actually the spelling of answer in African American is quite different from spelling of answer in, say, a Caucasian population. And that's actually one of our concern is how many flavors of answer ASO are we going to need to prepare. But again, right, if we have a regulatory system where spelling doesn't matter and we can approve en masse without worrying about the subtle spelling, then we are fine, right? Because making these things, a lot of us have ordered PCR primers, right? You just change the sequence, it's nothing. We should be able to make RNA medicine with the same ease and at the same cost as a PCR primer. And we have to put that as our goal, right? Yeah, thank you. All right, thank you very much. Oh.
I was kind of just echoing around those same lines actually, because with the Human Genome Project, you take multiple different diverse patients and you make this genome that's very diversified.
Yeah.
So using that same implications for your RN genome, I think would be very influential as well.
Yes. Right, since I took advantage of the time and kind of built the consortium before anything happens, right? And for the purpose that we can bring in an international group so that we can think about these exact issues at the very first step. Yeah, you can find me by email, so look for me. Thank you.
Wow.
Thank you, Dr. Chung. That was amazing. I'm double speaking here, I think. All right, we're entering the lightning talk portion of our symposium today. Please join me in welcoming Dr. Ankur Shah. Dr. Shah, you have 10 minutes, and my team will be right there flagging you.
Okay, thank you. All right. Just gonna put my own timer here. All right. And how do I get this to move? All right. So first of all, thank you to the organizers for inviting me, and thank you to Advance-CTR for the support in our work. So I'm going to be talking today about a very different topic, preliminary trends in on-site dialysis in skilled nursing facilities. And what we want to do here is quantify nursing home dialysis. These are my disclosures. I want to highlight Advance-CTR and NIH and IGMS for the support for us to be able to do this work. And so first I want to start with a little bit of background about why we're doing this. I'm going to skip right to here, which this is a policy change that happened in 2018. If you are a dialysis patient and you are in a nursing home, prior to 2018, you had a few options for how you would receive dialysis. The most common way that you would receive dialysis was the way that most people do. You would transport off-site to a dialysis unit. There were a couple other ways that you could do this. You could open a dialysis unit right next to the nursing home. And so even though it was administratively distinct, it was pretty much attached. Or you could get dialysis bedside in the nursing home. In 2018, CMS said, we're going to change this. We're going to allow you to open a dialysis unit inside of the nursing home. But we don't really know what has happened since. The literature on this is split in two ways. There's a couple of papers that have been published by one for-profit entity that specializes in this care model. But most of the literature on this is actually in the limitations and discussion sections of manuscripts. And mostly what it says is that you cannot identify this care model. And that's because CMS allowed it and regulated it, but they didn't really create a pathway to monitor it. And so what we've chose to do is try to identify this care model. And doing so required a bit of work. We had to start with 4 data sets. So we had to take Medicare data, take the Medicare beneficiary summary file, find dialysis patients in the Medicare data set, either through the ESRD entitlement or through looking at Medicare claims for dialysis. Then we had to link that to another data set, the nursing home minimum data set. This was to see when you enter and leave a nursing home. We had to take another data set of dialysis facility reports. And this gets a little bit complicated because interestingly to Medicare, these dialysis units don't exist. They are one unit with like a spoken hub model. So when you look for these dialysis units, they're not in the dialysis facility report. So we had to get a little bit roundabout to find them. And so essentially we had to use a combination of dialysis facility reports, nursing home facility reports, Medicare claims, the Medicare beneficiary summary file to be able to triangulate this population, which is why all the literature right now is basically limited to saying you cannot identify this population. And we were thankfully able to do that with the support of Advance-CTR. So we've created a cohort study. I'm going to be giving you just some descriptive results today. Oh, I'm already at 2 minutes. So these are inclusion-exclusion criteria. And we had to classify these patients in a little bit of a complicated manner, as you can see here. But to show you results, so the first thing I want to tell you is this population is described as either a hidden or forgotten population in most of literature. Starting, this is not even on-site. This is just monthly trends in skilled nursing facility utilization amongst dialysis patients. A very simple question that was last answered in 2005. When I wrote the preliminary data for this grant, I didn't even have a citation past 2005 of how many dialysis patients are in nursing homes. Turns out it's about 9%, and you can see the reduction in COVID-19 and the rebound, but not to baseline. And this tracks with more or broader nursing home utilization trends. And this is what's happening with on-site dialysis. And so most of the discussion of on-site dialysis actually usually happens on the other side of this room. It's when I'm in the audience talking to someone else and I say, you've seen a lot of people getting dialysis in nursing homes, haven't you? And we say, yeah. And then there's nothing in literature. And we're right. It has gone up by 212% since 2017 to 2022. We've gone from 8% to 18% of patients getting dialysis on-site in a nursing home. We asked a little bit about what's different about these patients. Where are they? What do they look like? There are huge geographic variations. And this validates because the group that's really, really tiny at the bottom is the West. And California does not allow you to open those dialysis dens that I described. In California, they have state policy that says you cannot open that dialysis unit in the nursing home. And so that is why California is much lower. The largest two are the Northeast and Midwest, because this is still very much a regional model. It's largely concentrated in Illinois, Maryland, and New York. And so that's why you see really huge variations, but you see growth across the scope. We were questioning about race and ethnicity variations. People who are African American have much higher rates of end-stage kidney disease. And one of the questions that I had was, is this care model being accessed by people with kidney disease, or is it being accessed by more lesser historically disadvantaged populations? And we see that the highest utilization is in African Americans and Hispanics. And so to recap, at 5 minutes on the dot, we show a sharp rise in on-site hemodialysis within skilled nursing facilities. This growth is uneven. There's substantial geographic, racial, and age-related variations. Comorbid conditions are very common. What does this all mean? Maybe next year we'll chat about comparative effectiveness and talk about some of the other populations that this is spreading into that need to be deduplicated out. So I am already out of time, so I will open up to questions. I just want to say thank you. There's a lot of people who are responsible to help me get here, including Advance-CTR, Dr. Rounds, the support from this group. So I just want to take one more second to say thank you to a lot of people.
And also to Brown Physicians Inc.
Yes.
But, so this shows it's hard to improve things if you don't have data.
Yeah.
What is the future of these data sources that you have been utilizing so effectively? Will they continue?
That is a really great question. I think that the data sources that we've been using have been put under a little bit of threat because there's been a change with Medicare that was proposed last year that may or may not happen, where instead of allowing us to own our own hard copies of the Medicare data, we would need to create a virtual, or we need to sign up for virtual access to their servers. And it is extremely expensive, not really doable on a career development award, mentored research award, or K. So that's one of the problems. The second problem is the United States Renal Data System, which is not what I used here, but which I hope to replicate so that they can take my methods and we can apply this more broadly. That is a federal data system that describes the epidemiology and outcomes of all kidney failure patients, is operated by the National Institute of, a contract from the National Institute of Digestive Disease and Kidney Disease to the University of Michigan. So we don't really know what's going to happen with the USRDS contract with NIDDK. So I think that my hope, I'll give you my hope, is that we will be able to create a pathway that others can replicate what we've done and that others can be more precise in their epidemiology of this population. But the two sources that we have used and the sources that others use that I hope to be able to help others replicate are all currently a little bit threatened. Thank you very much. Thank you for the opportunity.
All right, please join me in welcoming Dr. Thais Sao Zhao.
Good morning. Thank you for having me. I'm an assistant professor at URI College of Nursing. In case you never saw this is Point Judith. This is very Rhode Island, very local to us in Narragansett, and one of the 10 top ports in the US. So they had this huge infrastructure. I will tell you more later. But I want to thank a lot of people, a lot of support, but especially Advance-CTR because this was the very first project to work with fishermen in Rhode Island, and they bought my idea, very enthusiastic. So I was very glad and thankful for that. I couldn't have done this without the College of Nursing and local fisheries leaderships as well, because we all work together very closely. They brought me into their community, so it was very, very helpful as well. This is a huge topic in the US. Of course, this is the ocean state, right? But even across the nation, fisheries are a major source of income and a lot of productivity as well in terms of catch across species and across states. We have 2 recent reports showing the impact in Rhode Island right here, so you can have a sense of a small state and how much money and food does this brings to the table, right? So they call themselves the last hunters, which they really are because they are the only ones right now that are catching like wild things that we can have in our tables, and they feed a huge part of the state. And even with all that, they're still a very invisible force. So as long as they're working and bringing money in and bringing fishes and shrimp and all the things we love to have from the ocean, it's fine, and we don't know anything about their health. So there's no public database. There is nothing in Rhode Island or even nationally. So when we first got interested in working with that, we wanted to see, okay, so what is the problem? And we couldn't know because there was nothing there. So this was a very small pilot just to have like a photo of what's going on. So our goal was to examine the cardiovascular health, ideal cardiovascular health actually, and then quality of life, lifestyle factors, lived experiences, and productivity through catch, like really learning how much and how do they behave. So I won't have time to talk about the methods, but there's the study protocol is here. And I'm just gonna jump a little bit to our results. So we had 2 phases. Phase 1 was a focus group with fishermen and leadership to inform if our ideas were kind of in the right place. So we adjusted based on what they shared, we adjusted the survey phase that was informed by phase 1. So for the second phase, we have 59 fishers who respond to a number of questionnaires, and we were able to have some objective measures as well in 3 ports in Rhode Island. So for that, we had a whole year to do data collection because it was very challenging. I'm happy to share more about how recruiting is for this population later. But our main resource was Life Essentials 8 from American Heart Association, which allows us to use additional measures as well. So on top of all the health factors and behaviors you see there, we could add some very specific questionnaires as well to gather more information based on what they shared in the previous phase. And then we also had the secondary outcomes, quality of life and lifestyle, mostly because we wanted to add stress and behaviors related to substance abuse. So this are the results for phase 1. We had 5 main themes and 19 quotes that emerged from that. I have a lot of quotes to share, but a few there that I would love to tell you. They were very new to me. They sleep better in the boat, actually, that they do at home. They are very strong and tight community. One of the phrases that I heard that most resonated with me was that they say, this is not a job, this is a lifestyle, it's part of who they are as people, so it's everything they do. So they're really passionate about what they do, but they're getting very, very sick. And we need to do something as people who care for communities. So here you can see a comparison between considering of the 59 who are considered with ideal cardiovascular health using Life Essentials 8 versus those who are not. And which is a bit surprising. Of course, we used non-parametric tests. This was a non-normal distribution. So of course, there's not a lot of power in the test. And this is just a very initial exploration. But we can see, even being a very active profession, there's still there's a lot of room to improve. They're not ideally healthy. So despite the only thing that we couldn't find a difference was regarding age. Everything else, they are very, very different, especially substance use as tobacco and alcohol. And then for correlations, we were not expecting a lot of things. We found significant correlations for stress and hip circumference, which is not super intuitive to figure it out. We're thinking about binge eating and knowing what we know now about their eating habits. And then it could be like stress leading to weight gain, probably. But we need to know more about it. And the other one was systolic blood pressure and neck circumference, which is a very common and useful predictor of cardiovascular risk. So we have, what we can share now is a very preliminary picture of the situation, but there's a lot of, we could see there's a lot of room to act as in terms of interventions and how we can change behaviors and improve their health outcomes. Overall, what we see is that there's an overlapping in terms of behavioral, occupational, and structural risks, but at the same time, they're very open to receive help. So it takes a village, and we need to figure better ways now. Now that we know this little about them, we can at least start designing interventions to help improving these behaviors. So yeah, thank you.
Okay, so you need to know I just came back from the Mediterranean. There's a ton of fishing done there. Take this study over there, and I'll be your RA.
Now, let's go.
Questions? So nice to see you.
Thanks, Thais, for this really important work. As you kind of think about perhaps a community-informed intervention, and you talk about some of the unique personality traits and camaraderie, what are some things that you're looking towards and perhaps exemplars in other parts of the world that you may lean on?
In terms of designing interventions now?
Yeah.
Yeah, so there's a lot of problems that we could address right now, because I have a mentor who works very strongly in tobacco use. We are starting with tobacco for now. So we just designed an intervention to try nicotine pouches and compare that to not try to quit smoking at all, because if you never thought about it, international waters, they can do whatever they want because there's no regulations at all. So not only tobacco, but a lot of other things. But anyway, we decided to compare nicotine pouches with smoking to see if they're able to at least reduce a little bit the harm. But that's one thing for now. But there's a lot of other things, like eating behaviors are very poor. And they make jokes, like they know it's not good. So they know they eat things like candy, it's one of the preferences, and a lot of energy drinks. So there's room for doing a lot more. For now, we're working with smoking because we're thinking of the cardiovascular risk, COPD, and all the things that are coming with that, because the main reason, I guess, this relates to chronic disease so much is because they're getting older, and their siblings, their children, they don't want to keep doing this anymore. So those fishers, they cannot retire. So they're getting older and sick, and we need to find a way of coping with that. Does that answer your question? Okay.
Yeah. Again, I'm going to insist, a lot of smoking in the Mediterranean.
Today, we're living.
Super interesting work. I've got a couple of questions. One is, I'm curious how, when you did your focus groups, how that changed the work that you were interested in doing. You mentioned there were some surprises there. Yeah. And I'm also interested in now how you're using those folks that you engaged with at the beginning to help you understand the results that you've gotten preliminarily.
Yeah, thank you. Great questions. So for your first question, the main thing that was very, very new was stress. I mean, we could assume there's some stress, but it wasn't our focus. Like mental health or stress was not our focus in the beginning. And we decided we will have to do something about it. So we decided to measure. I think was the big thing. Other issues like trauma and orthopedic issues, a lot of pain. They live under a lot of pain. And they are like those type of strong guys that they just don't care about the pain. Just it is what it is, which is really not. So it's another thing that they're probably depressed too because of living with this pain for so long. So those were very new things for our group. And what could you repeat again your second question? Are you going back to them? Oh yeah. I did. Yeah, I did a lot already. Some things I feel like what we did, we went back to the commercial fishers of Rhode Island, which is led by they have this great leader. He has been in this position for decades. And he's just this amazing, charismatic guy. He felt that nothing was new, that he knew, like he knows his people. But he was surprised about the smoking a little bit because their average is higher than the national average. So he was impressed. And he knows what they do when they are overseas. And they stay there for long. Like trips can last up to 10 days. So they engage in certain types of behavior. So they were a bit surprised about some of the behaviors. Not all of them. He knew we had, and this was the first person that I called when I said, I want to do this. Would you let me in? Kind of conversation. And he was very open and happy because I was the first one to let's try to do this. So it was very a very good feeling of learning and sharing.
You're right at the doorway of the question I had.
Yeah.
Because, as you were describing these men, you said that it's not a job, it's a lifestyle.
Yeah.
And as you described him, it was very much a man's world. And all your photos had fishermen. And then I'm looking at you. So here's my question. Does gender matter? Did it matter that you're a woman, that you're not a fisherman? What really was the basis around which you built the trust? Where did the trust come from?
Okay. Thanks. That's a great question. And I think that was always my major concern since day one. I think the main reason that we were able to connect was this person, this leader, because he had a heart issue and he cried on the phone when I said, this is what I want to do. And being a nurse, I told him, I feel we have so many similarities because nurses are kind of invisible too. As long as they're working, we don't really care what's going on. So I started going there and just sitting and listening to the meetings and learning about their concerns and just being invisible, but there. And it took a long time until I was able to really join a meeting and be the one speaking and asking questions and making sure that whatever they told me was not leaving that room. And then the data collection was very long because you have to go, they don't come to your lab. You have to go there. They won't leave the vessels. So you have to go there. And sometimes we schedule times with them and we couldn't do that because the weather was amazing and they had to go. So it's a lot of rescheduling and just sitting there and being open. And I think I had great students helping me and being there too. And we did our best to be open and not judgmental about how they do. And I think that we were very lucky because they want to help. They want to receive the help. So once they see that we are going there just to offer something, I think they're happy to be seen. That was
Thank you so much.
Yeah, thanks.
All right, please join me in welcoming Dr. Luke Acuff.
Hi, everybody. I'm Luke Acuff. I'm a biomedical engineer. As you can see from my really lengthy subtitle there, I wear a lot of hats at a lot of different centers. Today, I'm going to be talking about work that we did at the Providence VA that also collaborated with the Cobre Center for Neuromodulation at Butler Hospital. So, in particular, our study's interest is in depression, primarily in finding biomarkers of depression. Of course, depression is one of the more slippery disorders, especially in psychiatry, when it comes to finding reliable, repeatable, and reproducible biomarkers. However, there's a lot of, obviously, a lot of really big upside should we be able to find biomarkers that we can see in neuroimaging. Our particular interest as an engineer that works primarily with clinicians is improving treatment. There's a lot of neuromodulation work at VA and at Butler Hospital and at Brown. And with good reproducible biomarkers that can both help us assess treatments for depression as well as inform new ones for purposes of targeting and things like that. So in particular, we're looking at machine learning. A lot of the neuroimaging data that we have is very large. We have really, really large data sets that we're working with. So machine learning helps us break that down and identify patterns that aren't really visible to the human eye. In particular, we looked at EEG coherence. So EEG, we throw a cap on the head. We have 64 electrodes on the cap, and we want to know how connected, roughly, different electrodes are to each other. We put that in IDS-SR symptom domain scores, which quantify how severe depression is and also what sort of depression a patient might have, into a machine learning algorithm that's referred to as LARS. So we start with feature extraction. This was data collected at Providence VA. So this is a pretty small data set to start. We're collected from 19 veterans with depression. We extracted coherence between all channel pairs, as I mentioned a moment ago. That is a measure of functional connectivity, so the idea that these areas are connected in some way, at each major frequency band, so delta, theta, alpha, and beta in EEG for our purposes. This is a really large, dimensionally large type of data. 64 by 64 channels ends up with something like 2,000 pairs, and you've got that at each frequency. So to make this reasonable not only for us, but also for our model, we dimensionally reduced it with PCA. And then we paired those dimensionally reduced factors that still represented the larger picture with our total depression severity scores. Again, the total score, or broken down into subdomains of depression. So the mood aspect of depression or the anxiety aspect of depression as established by Wardenar's factor analysis. So we feed these into our large model to see if that brain data can tell us, without knowing per se, what sort and what severity a person with depression might have. So before we get into results, one thing that we have to talk about with machine learning is model validation. Probably the biggest challenge in machine learning is overfitting. You can give a lot of the more advanced machine learning models today total garbage, frankly, and it will tell you a really beautiful picture about what all the data means and try to please you and give you something that is completely and utterly unreproducible and meaningless. So what we have two ways to combat this. The first is leave one out cross-validation. All of our reported scores, when we talk about how well our model performs, we are referring to cross-validated scores. So we run the model multiple times, leaving out a participant each time and validating the model on that left out participant. You can average that to get a more reliable measure of how well the model works because it's not measuring against trained data. And we also optimize one of our modeling dials, so to speak, regularization, to optimize that value. The other thing that we do, and this is probably the biggest takeaway from the talk today, my very brief lightning talk, is using real and quote unquote fake data to check against overfitting. So you can see in these plots here, those red lines represent how well, that's got R squared on the bottom, how well our model performed using the real correctly correlated data. The gray bars represent model performance on shuffled or permutated data. So what we've done is we've taken our IDSSR scores, the clinical sort of representation of depression, and shuffled them with respect to how they map on to our brain scores. So if the model performed really well with our shuffled scores, that would tell us that it's overfitting, right? We're giving it bogus data and it's painting a picture anyway. But we can see, especially in terms of anxiety in our anxiety subdomain, the model performs really well with real actual data and really poorly with fake data, which is promising for us. That tells us that we're picking up on something real, even in a really small data set. And this might be one of the, this tells us this is a correct region of machine learning to move forward and to investigate other things like response, larger data sets, open source data sets, et cetera. In any case, we found our best results in the beta band. So for all of you EEG researchers out there, I know beta is a hot topic right now, so happy to talk about that. But here's our sort of predictive model. And this represents our coherence network weights. I wouldn't try to interpret this too heavily. EEG, that's one of the limitations of EEG is that it's not really super source specific. So I could say broadly that we're seeing a recognizable pattern of quote unquote frontal beta coherence, but what brain areas that corresponds to exactly is a little bit beyond the scope. So in conclusion, the primary takeaways are that anxiety, funnily enough, in a depression cohort seems to be the most readable to our machine learning model, in particular in frontal beta band coherence. But that's really promising. That gives us a very specific area that we can pick up in the brain and correlate those signals to sort of the character of the depression the person has. We also have really promising evidence that the LARS model has some resistance to overfitting. It performed well on real data and poorly on fake data, and that's really promising for us. Our primary challenges right now, the first is limited interpretability. So EEG, as I mentioned earlier, it's not fMRI. You can do some things like source localization to sort of hone in on different areas that might be more affected, but you can only take that interpretation so far. And of course, it's a small data set. And the other thing is that this comes from a highly homogeneous sample. So I mentioned earlier this was 19 veterans at VA. Most of them have comorbid PTSD. They're primarily white males. And one of the downsides of this is that while we might be able to identify a really salient signal in a very generally similar data set, when we try to upscale this to larger data sets, as we've attempted to do with some of the data at Butler and the CoBrief at the Center for Neuromodulation, we start running into issues, which may tell us that this is an area where individualized approaches are appropriate, especially with respect to depression. And we might need to work on fitting models to subtypes and subscores and even populations that experience similar types of depression in order to better understand biomarkers and better generate biomarkers that can inform treatment and diagnosis down the line. So I'll go ahead and take questions.
So EEG is a much easier and less invasive and time-consuming and expensive technique than some of the other imaging techniques that you mentioned. Do you see this becoming a more widely used tool to individualize the type of depression that someone is suffering from so that you can change therapy accordingly?
Absolutely. So you raise a really good point that EEG is a much cheaper and more accessible form of neuroimaging. fMRI is great. I do a lot of fMRI research. The problem is that if you want to take TMS, which is something that our clinic is really interested in, a form of neuromodulation, you can't get these people to come in. You can't get all of your patients to come in and get a scan. It's too expensive. It takes too much time. And it's already hard enough to get people to come in that are suffering from severe depression, day after day to get the treatment. EEG, on the other hand, is quickly, I'm sure my colleagues in the room have often struggled with getting the cap to work. So I can appreciate that. But compared to other techniques, it's much faster and cheaper. And the techniques for analyzing and breaking that data down are getting better. So I see it growing primarily because it's necessary to have quick, fast, and available neuroimaging techniques to inform this sort of work.
Amazing. Thank you very much. Round of applause.
All right, please join me in welcoming Dr. Kwesi Lillard. Excuse me.
All right, thank you for having me for this presentation today. My name is Kwesi Lillard, and I'll be discussing utilizing RNA sequencing to identify markers of site-specific bacterial infections. So COPD is the fourth leading cause of death in the US and worldwide. It's a progressive airflow obstruction resulting in chronic respiratory symptoms, from airway inflammation to alveolar wall destruction. Pneumonia is the leading cause of death worldwide and the leading cause of infectious-related death. So the two of these conditions carry a significant mortality rate together, and due to the susceptibility COPD patients have to pneumonia, early treatment and diagnosis leads to improved outcomes, so it's imperative to accurately diagnose and treat these patients. So now, what is RNA sequencing? RNA sequencing is when we isolate all the RNA from a patient blood sample, and we sequence it and convert it to 150 base pair fragmented sequences. These are then analyzed computationally to identify, for our purpose, pathogens causing disease. This method is promising because cultures which aid in diagnosis can be slow in range and accuracy, and sputum cultures can range from 50 to 80% in accuracy. RNA sequencing can give us these results in less than 5 hours. So given this, our hypothesis is that utilizing RNA sequencing, we will be able to identify genes specific to pathogens that will vary by site of infection. So this was first seen in a paper by DeMello. This paper looked at the mouse model, and they followed Streptococcus pneumoniae and how it was colonized with the nasopharyngeal swab, and they performed RNA sequencing following the mice, and they were able to determine what genes were specific to colonization versus dissemination to other organs. So there's two figures here. Figure A is looking at the genes specific to the bacterial expression, and then the green circle looks at the colonization just in the nasopharynx. But then when you look at the boot dendrogram, it confirms that the clustering of Streptococcus pneumoniae specific genes varied by the anatomical location that the genes were expressed. So overall, the results showed that the Streptococcus bacteria was able to change its transcriptome to adjust to the host environment, and again, varying by anatomic location. So we took 38 ED and 62 ICU patients with Klebsiella pneumoniae, Staph aureus, and Pseudomonas aeruginosa infections, and they also vary by site of location from urine to sputum and bacteremia. And we took their whole blood samples and sent it for RNA sequencing at 100 million reads per sample. And we took these sequences that were obtained and aligned it to our pathogen of interest so that we could identify novel targets. So here's just some tables showing that the sequences that we found specific to each pathogen's environment. So we have the bacteremia group, the sputum group, and the urine group. And just in bar graph form, again, it's just representing the different groups of bacteremia. The blue bar is just noting the total amount of sequences that we identified. The orange is notifying the ribosomal RNA. And then the green, which is a little harder to see here, just in the range of about 30 sequences, were the mRNA sequences that we identified. Again, here as well, in the sputum group, we have the same kind of breakdown, with the blue being the total reads, orange being the rRNA, and then the green being the messenger RNAs that we identified. In this group, we also identified other novel targets, like novel or unannotated targets, like novel regions, other types of biotypes of RNA, like tmRNA and tRNAs. And then going to the urine group, we also found similar sequences where again, total sequences are in blue, and the other biotypes are in orange, green, and blue. The lesser sequences found in the Staph aureus group here on the left, this kind of reflects that the sample size that we had for those patients were a lot smaller than the other samples. It's important to also note that given all these sequences that we obtained, one sequence wasn't found in any other group. So the sputum groups were specific to the sputum reads. They were not in the bacteremia groups or the urine culture groups, and that they were also not in the negative control patients. So to make sure that we have a sequence that's specific to our pathogen of interest, we then take the sequence and we blast it in the NCBI database and see that this sequence, for instance, had 99% specificity as Staphylococcus aureus. And this is just an overall breakdown of how we're able to find site-specific markers varying by pathogen and location of anatomic location. So clinical implications in chronic disease states, especially diseases like COPD, there's an increased risk of mortality. Earlier infection detection leads to improved outcomes. Improved localization leads to better informed personalized care and administration of appropriate antibiotics, which will also help us combat this global health crisis of antimicrobial resistance by identifying the causal agent. And again, I just want to take this time to also thank my lab and for everyone for the opportunity to be here today.
Nice, thank you.
Thank you, Dr.
So, how far away are you from this replacing routine culture and sensitivities, which take, sometimes 2 or 3 days, sometimes longer, and significantly decrease the the likelihood that you're going to be treating with the appropriate antibiotic?
Yeah, thank you, Dr. Rounds. It's a very important point. So blood cultures, sputum cultures, a lot of these, you have to culture the bacteria, so it takes days to get our pathogen of interest. Sometimes you can have contaminants. So there's a lot of factors that go into play. For us right now, we just finished the computational side. So we identified these target sequences that we're going to design primers from. And our next step is to move over to the wet lab and test these primers on our pathogens to confirm, or our patients with these confirmed pathogens, to ensure that these primers are correctly hitting the desired target of interest.
A few more steps.
So a few more steps. But this week we're starting to test things, so hopefully we have some preliminary information soon.
Thank you.
Thank you.
All right, please join me in welcoming Dr. Ryan Close. And since you're last, you got the benefit of a little bit extra time here. So,
Yeah. Hopefully we don't need it. Is this, how's this click? How's this? Okay. My name is Ryan. I'm a primary care doctor and epidemiologist with Maine Health. And I have a research focus in urban-rural disparities. And we're speaking today about mobile phone auscultation for the detection of structural heart disease, which I hope to be a step forward in addressing those disparities. So I live in the state of Maine, which is the oldest and most rural state in the country. And so naturally, I and my colleagues are worried about diseases that have age-related prevalence and urban-rural disparities. And that includes cardiovascular disease and structural heart disease. So today, if you are a rural patient, you are more likely to die from cardiovascular disease than your urban counterpart than you were 20 years ago. So all those developments and improvements in cardiovascular health have been largely concentrated in urban settings. Rural patient health has improved, it just has not improved nearly as much as it has in urban settings. And that has to do for a lot of reasons: access to primary care providers, to diagnostics, and to specialists. And so the problem before us is, how can we use novel technologies and methods to improve the detection of structural heart disease and cardiovascular disease in order to reduce related mortality? So it's this paradigm I'm hoping to shift or expedite, getting the patient from the far left of the screen through the long, lengthy process of PCP or frontline clinician, to the eventual test, to the eventual cardiologist, and then eventually treatment someday. And what we hope to demonstrate is that by using sound recordings from unmodified mobile phones, can be used to develop an accurate model fitted to echocardiographic gold standard data for the detection of structural heart disease. So this is how we do it. I love a good diagram that says everything that I can't say in words anyways. So, and I'm a little terrified there's a biomedical engineer in the audience, so because I'm not a biomedical engineer. So this is what we did. We approached a bunch of patients who were getting echoes as part of their routine standard of care. We then grouped them based on their findings, whether they had aortic stenosis, our first model of choice, other structural heart disease, and control patients. We collected sound recordings from them in various sites of their chest, collected by our team as well as the patient themselves, then did a huge chart extraction to get basic comorbidity data. Then the model making. So we do not use machine learning. We do not use artificial intelligence. We use a test and train model that applies nonlinear dynamics, which is a physics-based mathematical approach that understands the dynamics of how the heart is functioning. On your left, you will see this is sound as you and I understand it in the frequency domain in unidimensional. When you transcribe sound through nonlinear dynamics, you get what is on the right, and this is called a phase space. This is what sound looks like when you analyze it using nonlinear dynamics. We then perform PCA and KS analysis to extract all our various features. In a 30-second recording, you can get over 50,000 individual features in one sound clip. We then use combinatorial analysis to identify which combination of features are most indicative of your specific disease state. Currently, AS is what we're most concerned with, and we just map those features, 3 or only 4 of them, using logistic regression to get our BA chart of true positives and negatives, true and then false positives and false negatives. So we'll skip through the results of what who the patients are and get to the model. So MLE and D-core, these are nonlinear dynamic variables, confirmed that all our recordings were chaotic and of low dimensionality. KS analysis allowed us to identify and extract our features of choice, and we created 3 predictors. And through our combinatorial analysis, we identified 2 specific acceptable models that had performance characteristics that we enjoyed. And as you can see in the middle, our test and train sets had sensitivities and specificities that ranged from 92 to 100% and 82 to 89% respectively. So we are hoping to shift this paradigm, one in which we go from this lengthy model to perhaps in the hands of a PCP or a frontline clinician using 30 seconds of sound alone can diagnose or screen a patient for structural heart disease. You can even imagine this being applicable in a telehealth model. Because mind you, it kind of breezed over it, but we have a model basically that is as predictive as echo for detecting aortic stenosis of 89 to 90% with only 30 seconds of recording. Next, we're going to complete these AS models. We're now mapping it to aortic valve area, seeing if we can create a model that maps not only the presence of AS, but also how severe the AS and what the valvular size is, which is important for intervention and how quickly they need to see a cardiologist or potentially get valve replacement. And then we're going to repeat these models for mitral regurgitation and then hopefully get funding. So thank you. To the biomedical engineer in the front. Brace yourselves. Whew. All right.
Yeah, you just get the rural researcher instead. So I think this is super interesting. I, as a former division chief that did a lot of work with telemedicine and with in looking at a lot of those kits, those carts come with integrated auscultation for some of them. And speaking of somebody who goes out into rural Nebraska, not infrequently, we're not, maybe not as rural, but we got a lot more like frontier space than you guys do. So I'm interested, are you working with any device companies? I love the smartphone piece, but thinking about this from a primary care standpoint, have you thought about any partnerships, strategic partnerships with some of those telemedicine companies that have those built-in carts?
Yeah. So the reason we've gone the smartphone route is that we can use any phone on the planet. This can be downloaded to the patient's phone or used by an individual's phone. It's the phone is not fancy at all. It's just a regular phone. So it could be an application that's downloaded to a phone that's anywhere. So we haven't approached device companies because in an attempt to address rural disparities, we like the ubiquity, the fact that there's billions of phones on the planet already.
Don't disagree. Do you think you'll get 510K approval or FDA approval to use this on a smartphone device?
So a discussion we're thinking amongst ourselves is if it's a screening tool, not necessarily intended to change clinical course, but just to inform, it might not need the degree of FDA approval, as you would say, for echo. So these are those commercial sort of viability plans are really in the hands of people who hold the IP. I'm just the guy who wants to see who it can help. So we'll see. TBD.
I'm thinking downloading IRB problems, maybe.
Yeah.
Tremendous work. Thank you so much. Can you tell us more about the IP? I mean, I know you can't reveal everything, but but somebody has intellectual property on this.
Correct.
Okay.
Yes. I can tell you someone has it.
Not you.
Yeah, well, no, definitely not me. I think what's nice about nonlinear dynamics is, and this application has been used for COPD, pneumonia, heart failure, volume overload, is that it's analyzing sound to tell you the function of an organ system, not the sound of an organ system as you and I understand it. And that it's using data and sound that is far beyond what our brain can really interpret, and has, I think, really vast implications and applications because knowing the dynamics and the function of something goes far, including infrasonic, goes far beyond what we can hear. And so there are going to be applications of this technology that are going to really expand how we can monitor people's disease processes, I think.
All right, so that's kind of a lead into my question, which is, why did you pick aortic stenosis? Versus, which certainly, no question, it can be devastating to individuals that have it, but it's relatively rare, versus aortic aneurysm screening, which we know there's important evidence for.
It was largely coincidence. Unrelated to this, I do structural heart disease work in community-based settings in the West. And the AS is really just the first of our whole model of structural heart disease. So we've already done feasibility for volume overload and heart failure. So we're going to add structural heart disease, which is going to be AS, MR, and other. And we've already done COPD. So it's really like a cardiopulmonary platform. And AS is just the first model that was built out of this study.
Right here.
Sir.
About maybe 20, 25 years ago, I was working with colleagues at Mayo Clinic, and they would come down to work with us in their inner city barbershops. And one year they brought echocardiogram, and it was the size of this computer here. But they said there was a time when that machine would have been in a room.
Yep.
Okay, so 20 years ago, the echo comes out of a room and into a laptop.
Yep.
Now I want to make sure I'm hearing you right. Are you telling me that now I got something the size of this phone that's functionally the equivalent of what I could get with an echo? Is that the lead here?
It could never be better than Echo because our modeling is based on mapping the sound data to Echo. So it could be as good as Echo, but not better than Echo.
Okay, so perfect need not be the enemy of the good. In inner cities where they may be urban, but basically you have lack of access,
Totally.
just like in rural,
Yeah,
this could make a difference, yes?
that's our goal.
And I could literally be in a barber shop or a beauty salon and not in a hospital, and at least get a signal and say, hey, you need to see somebody.
Mm-hmm.
Really?
Yep.
Right now?
Yeah.
Wow.
I'm with you. I'm thinking we're skipping Medicaid, Blue Cross, going right into any.
Yeah, I thought this was totally fascinating. As an engineer from rural Tennessee originally, I really resonated with a lot of this work, and it's signal processing, right?
Yes.
So I guess my primary question, because we worry about this all the time in our end, you're getting data from phones that, as you said, could be variable conditions, things of that nature. What sort of cleaning and pre-processing do you have to do to that audio data so that you don't end up having your model overfit noise or, you know, otherwise, you know, you might pick up on poor phone quality being linked to something, you know?
So it's interesting. So the phone microphones are excellent. And they're highly sensitive. They're very fast. They have a broader range than the human ear, and they pick up infrasonic sound, which is most of the human body makes infrasonic noise. Good for us, because if you could hear your heartbeat every second of every day and the whoosh whoosh it makes, you'd go crazy. But the phone's mic can pick that stuff up. And even though we can't hear it when we play it, the data's there. When you run these, when we went through the waves, they're very smooth. They've done, when we've given the model fake AI generated noise, the model can detect the difference. And those waveforms get jagged. And so it's very good at slicing out exactly sort of the dynamic of whatever it's listening to and interpreting that, which is excellent in our case. Though the key importance is you just have to have the mic on the chest, because if it's air, then it doesn't work at all. But in our study specifically, we gave the phone to the patient, the subject, and we said, record yourself. And their recordings were identical to those collected by study staff, which was important for a scaling up process for us.
That's fascinating. Thank you.
Oh, you want? Oh, I'm so sorry.
Okay.
No, great talk as well. You mentioned like starting with aortic stenosis. I guess on your end, when you guys were coming up with this, you mentioned that the technology is so that the ultrasound itself will pick up what a clinician might not hear.
Correct.
So I guess clinically relevant, because usually when they're more symptomatic, it's more audible. So I guess how many if someone has an audible murmur and they're symptomatic, then I mean, odds are they're probably already kind of getting treatment. So what number of patients is this going to be affecting, I guess? That the GP or the PCP wouldn't be already able to diagnose and treat?
Yeah, that's a great question. I think I would say, speaking in large generalities, the physical exam has been depreciated over time. The ability to pick up an actual murmur is low. Like, you might hear it, but like, you know, not every time you listen to the chest, the person with the murmur always has the same degree of the murmur. There's also low flow, high grade, or variable grade aortic stenosis. And this is just talking about AS. And the whole point of catching AS is to catch it early, because by the time they are symptomatic, morbidity skyrockets. So you want them, that's part of the reason we liked AS, is that it's intervenable, and you want to intervene before they have symptoms or it's severe, because by then you're actually too late clinically to make a huge impact on their morbidity and mortality. And what will be exciting about the dynamical approach is we'll be able to sort of observe dynamics of hearts in ways that we can't hear. Just as a side note, they already did this for EF, and when you take the predictive features that are most indicative of a low ejection fraction and you put them back into a device to say, what does this sound like? It just doesn't sound like anything. But the dynamic model can hear what low EF sounds like, even though we cannot hear what a low EF sounds like. And that's what's crucial, is that it's thinking of sound in a completely different domain.
So I'd like to put in a bid for other sounds that emanate from the chest.
Yeah, and it doesn't even have to be from the chest.
Well, I'm thinking particularly about breath sounds. For the last 2 years, I've worked with a class in biomedical engineering here at Brown, trying to stimulate the students to figure out how to use sonography to differentiate between crackles, rhonchi, wheezes, and normal breath sounds. Have you thought about, thinking about other sounds?
There's a paper coming out next week that I'm on that is this study for COPD. Really? Over 90, using 10 seconds of egophony and 30 seconds of an axillary sound in a composite model, had a sensitivity and specificity of over 90%. For the detection of COPD. Next step is recreating the GOLD criteria. So next week, coming up. Yes?
If we think about AI use and some of the comments about things other than cardiovascular disease, other than structural, we know that AI is being used in ablation with incredible precision. Have you thought about the addition of AI to perhaps promote earlier detection?
So AI and the biomedical engineers in the room can speak, right? Has been quite successful with images and image detection. We have colleagues who've used AI with screenshots of EKGs, for instance, ECGs, but it's had a hard time with sound, and that's because it is still looking at sound from its frequency domain. And as I was trying to display earlier, sound is multidimensional. And even if you run 1 million samples of a unidimensional sound frequency through AI, could overfit, or it'll have shortcomings. And so we only needed 250 patients to accomplish this. I do think there will be a place for AI and machine learning to do the combinatorial analysis, because that ends up taking like 9 days on a computer. Doing the logistic regression. And so I think there will be a space for it to come in sort of at feature extraction and analysis, but not really for the math. The math wasn't possible till the 1980s with supercomputers, but the mathematical principles were already there with Taken's theorem.
I'm thinking of all the data points that you mentioned, so even just throwing those into some type of
Yep.
analysis and seeing if there's certain phenotypes.
Yep. It would be able to do exactly. Once the math is applied and the features are extracted, I think there's a place for machine learning and AI to basically do the analysis a lot faster than is currently done.
I just have a comment, and thanks for this. And it's Thais, I think your fishermen need to be in contact with Ryan's devices, right? So.
One more question from me.
Yes.
What are the numbers you're targeting to recruit?
So for a phase 1 feasibility, we only need about 65 persons per group. So it depends how many models we want to build. So for this one, we only did 3 groups. We'll be making a mitral regurgitation model next, and then we'll try and see how well a structural heart disease other model looks like, and then healthy hearts as well. So it sort of depends on which model. Next phase 2 though is probably 1,000 patients, multi-center sort of style, looking for collaborators.
One more. I'm sorry. Communities. What communities are you targeting? Are you looking?
So Maine is not the most diverse state. It's like top 3 whitest state in the country. We are, diversity is important for obvious reasons, because there could be, something in the dynamics of hearts. But, the engineers with this unlike genetics, right? I mean, there's a thought that the dynamics of the heart is somehow agnostic to the other demographic features of the patient. Obviously, the demographics of the patient affect inflammation and dynamics, but the dynamics are what they are. The patient has or does not have heart failure. It's sort of agnostic to its cause, if that makes sense.
Thank you so much. Sorry, Sharon, did you? I'm just doing some housekeeping announcements. Thank you.
Great, thank you.
I really appreciate it. If you couldn't tell, you're talking to the world's greatest patient recruiter right there. So.
The RA in me, the recruiter in me is thinking, but how? I'm gonna do the IDs. How are you gonna put in, stock them with the nurse? Yeah. I'll do much more.
Thank you all for joining us this morning. So we're going to head into our poster and lunch session. Lunch is being set up right now, but the posters are ready to go, and we have some fabulous presenters out there for you. If you're an EAC or IAC member, we'd love to have you join us by the staircase in the lobby. Brown communications team would love to get a group photo. And we'll be back here at 12 o'clock. We'll start our next session of lightning talks. We have some amazing presenters. And then also Dr. Donald Neese will present our second keynote on engaging PBRN practices and patients as partners in chronic disease prevention and treatment. Thank you.